Hello, everybody. Here I am again, coming with a new topic, which may also help you. This is a new topic from enzyme section, Unit 3, Grade 11 Biology, of course. This may help you in your genetics, molecular genetics, Grade 11, Unit 4. We have already started, and therefore, please study. DNA modifying enzymes. It talks about those very important, crucial enzymes which modify the DNA in the process of gene expression, the central dogma of molecular biology, including the replication process. Therefore, let's go. And the DNA modifying enzymes. DNA modifying enzymes are specialized proteins or specialized enzymes that catalyze chemical reactions to modify the structure or sequence of DNA, playing crucial roles in the process. The processes like replication process and repair, DNA repair process, and gene regulation and gene expression process. Therefore, they are very important in the modifying DNA so that we can control the process of replication, the process of whenever there is breakage, repair, and also regulation, gene regulation. Let's go. The first category, types of DNA modifying enzymes. The first category in this DNA modifying enzymes are the DNA methyltransferases. As the name indicates, you see, these enzymes transfer methyl group, CH3, methyl group. Methylation is when we add methyl group to part of DNA or gene. Methylation, methyltransferases. Therefore, let's see. These enzymes add methyl groups to DNA, a process known as DNA methylation. And this modification by addition of methyl group can regulate gene expression by affecting the accessibility of DNA to transcription factors. You see that part of DNA to which methyl group has been attached will not be easily accessible or transcription factor. So unless transcription factors come and attach to that region, there will not be transcription. Therefore, they block the gene temporarily. They block the gene from expression. Gene expression is temporarily, or of course in some genes permanently, by methylation they are shut off. The second group, aberrant, in this case, let's finish. Aberrant methylation. You see, whenever there is abnormal methylation or not proper methylation, aberrant methylation patterns are often associated with such disease like cancer. There are this abnormal methylation in genes or part of DNA means you can't add methyl group properly, so that you can shut off that part of the gene, so that transcription factors will come attached and there will be expression. This is a feature of this feature. Uncontrolled cell division means uncontrolled DNA replication, which means, of course, cancer. The other group, DNA helicases. These are the hydrogen bond breaking enzymes between the complementary bases between G and C, between A and T in double-stranded DNA molecules. They break the hydrogen bonds to unzip or split open the two strands, to split the two strands during DNA replication as well as during transcription, part of the gene expression, of course. These enzymes unwind or unzip, you can say, the DNA double helix, separating the two strands to allow for replication and repair. Repair is also part. When there is part broken, you have to unwind the DNA, the complementary hydrogen bonds between the complementary base pairs, so that the broken part can be maintained. They utilize energy from ATP hydrolysis to break hydrogen bonds between base pairs. You see, this utilizes energy to break down the hydrogen bond between the complementary bases. And the third group, DNA topoisomerases, this work on the topography of the topology of the DNA molecule. These enzymes manage the topological state of DNA by introducing or removing supercoils. Of course, they also introduce supercoils. If need be, they also just remove the supercoil, especially whenever there is helicase splitting the hydrogen bond and moving forward. If there is supercoil at the two ends, then topoisomerases cut either one strand or both strand and rotate and reattach that area so that it can remove the supercoiling. They are essential during DNA replication and transcription to relieve torsional strain. You see, whenever torsional strain, that is a supercoil, produces torsional strain on the DNA molecule, which may damage the DNA. So topoisomerases come and cut either one or two strands and rotate it and reattach that DNA so that they remove the torsional strain or the supercoiling in the DNA. And the third, I think these are the fourth group, DNA ligases. These are enzymes join DNA fragments by forming phosphodiester bonds, crucial for DNA replication and repair. Of course, these are very important to bring about repair, DNA repair workers, DNA ligase, DNA repair workers. Whenever there is damage in the DNA, broken phosphodiester bonds, they can seal that bond by using energy ATP. They build phosphodiester bonds, and that's also very important for replication process to join broken fragments of newly synthesized fragments of DNA, especially in the lagging strand during DNA replication. The lagging strand is produced in fragments. These fragments are finally joined by DNA ligase. They seal nicks in the DNA backbone, ensuring the integrity of the genetic material. Yes, genetic material. There should not be fractured or broken part of the backbone. The backbone is made of sugar phosphate backbone. The nucleotides join through phosphodiester bond. Therefore, whenever there is breakage in these backbones, the maintenance worker is by joining the broken nucleotide by phosphodiester bond. The maintenance worker is DNA ligase, of course. The other enzymes are DNA polymerases. This is one group, DNA polymerases. You know, the name indicates these are construction workers. These enzymes synthesize new DNA strands by adding nucleotides to a growing chain during replication. Really smart, super smart enzyme, except that it has two Achilles here, or two drawbacks. I will tell you about the mystery of this name. Of course, we can call it, this is very huge multimeric enzyme, very huge, not one purpose, just not building purpose. It builds, stops, moves back, and proofreads. That means checks for error, whether it has made error or not. Whenever there is error found, it will correct that error. A really smart enzyme. Therefore, this enzyme synthesizes new DNA strand by adding nucleotides to a growing chain during replication process. They also play a role in DNA repair. You see, they serve as just DNA ligase, repair by excising and replacing incorrect nucleotide. I've told you, incorrect nucleotides are cut out and replaced by during the proofreading activity of DNA polymerase. Therefore, DNA polymerase to have part which works as DNA ligase. It can seal, cut out, and join. Okay. The other group of enzymes are restriction enzymes. Look, restriction enzymes. These enzymes cut DNA at specific sequences, which is vital for genetic engineering and cloning. They cut DNA at specific sequences. Of course, when they cut, they leave sticky ends. They cut the double strand at specific region, but at the two tips, some 5 or 6 nucleotides are left without having partner in the other strand. That means sticky ends. They are free, not bound, like A, D, A adenine, guanine, cytosine, thymine, not partner. The other strand is missing there at the tips. These tips, where there is only one strand, are known as the sticky ends. Therefore, whenever restriction enzymes cut DNA, they cut DNA by leaving sticky ends at the two tips of the broken out DNA, the cut out DNA. And they can be used to create recombinant DNA by cutting and pasting DNA fragments. You see, they are very important to cut out the gene that you want from a DNA, donor DNA, so that you can transfer this cut out DNA into the recipient organism, so that you are performing genetic engineering. Therefore, they are very essential tools of genetic engineering to cut out the donated gene from the donor organism, and you attach it by, of course, using DNA ligase to attach into another organism's DNA, so that the organism that has received the new DNA, known as genetically modified organism, so it can produce the product of that gene if given suitable environmental condition. Therefore, restriction enzymes are one group in this regard. And last, nucleases. Look at these enzymes, nucleases, either exonucleases. These are specific to cut DNA from the two ends, or endonucleases, which love to cut at the mid portion, middle acting endonucleases. These are enzymes which cut DNA. These enzymes degrade nucleic acids by breaking phosphodiester bonds, the covalent bond that holds nucleotide together. And they can be classified into exonucleases and endonucleases, depending on whether they remove nucleotides from the ends or cut within the DNA strand. Therefore, those endonucleases cut the DNA from within at the mid portion, whereas those exonucleases cut the DNA from the tips. Therefore, these are DNA modifying enzymes. So far we have seen. Therefore, look, these are very important. Don't forget, please. Thank you very much. Please don't forget subscribing, liking, and sharing my videos. Please also try to put your comments. Your comments are welcome. Thank you. I will come another session by another program, another lesson. I'm sure probably may come with worksheet, of course. Thank you very much.